Good morning. In this video, we will be discussing about the biochemical importance of HMP shunt pathway. HMP here stands for hexose monophosphate, which is nothing but our glucose 6 phosphate. Normally, the glucose 6 phosphate, which is formed from glucose, will be oxidized in glycolysis and it will be utilized for ATP production. So this is the normal or the major fate for glucose 6 phosphate. But sometimes in some cells it may be diverted for an alternate pathway to produce NADPH and pentose phosphates. Okay, so such an alternate pathway which is undergone by our glucose 6 phosphate or the HMP is called as HMP shunt pathway. Since pentose phosphates are produced in this pathway can also be called as pentose phosphate pathway. Okay, so this is the definition for HMP shunt pathway, and an alternate pathway of glucose 6 phosphate to produce pentose phosphates and NADPH is called as HMP shunt pathway. Now we will see the functions of pentose phosphates and NADPH produced during this reaction. First, we will start with pentose phosphates. One of the major pentose phosphate formed during this pathway is ribose 5 phosphate. Actually, this ribose 5 phosphate is the starting material for both purine as well as pyrimidine nucleotide synthesis. Okay, so these purine and pyrimidine nucleotides in turn, they will be utilized for all the nucleic acid synthesis, both for DNA as well as RNA. We need ribose 5 sugar. Okay, in DNA it will be again reduced to deoxyribose, whereas in RNA as such the ribose sugar will be utilized. Okay, but for both we need ribose 5 phosphate. So thus our HMP shunt pathway becomes very important in any rapidly dividing cells like blood cells, skin and GI mucosa. There you can see large quantities of glucose 6 phosphate diverted for HMP shunt pathway. Apart from this DNA and RNA synthesis, ribose sugars can also be utilized for the synthesis of ATP and GTP, which acts as energy currencies. They can also be used for the synthesis of various vitamin coenzymes like NAD, FAD. All these things contains ribose sugar. Then apart from that, it is also used as a second messengers like cyclic GMP and cyclic AMP, etc. Okay, so thus these are the major functions of pentose phosphates, and for producing all these things, HMP shunt pathway is very critical. Okay, next coming to NADPH. So NADPH, it becomes the major source of a reducing equivalent for many reductive biosynthetic reactions. Okay, so this NADPH is very much essential for the synthesis of fatty acids, cholesterol and steroid hormones within our body. Okay, various reactions in these synthetic pathways, they require NADPH. Okay, the many reductive reactions takes place during the synthesis, and for all that reductive reactions, that reducing equivalents are donated by our NADPH only. Okay, so apart from the synthesis of these compounds, NADPH is also needed for various other processes like free radical scavenging, detoxification process, bactericidal activity of macrophages, conversion of methemoglobin back to normal hemoglobin, and for nitric oxide synthesis. Okay, so now we will see in detail how our NADPH plays a major role in all these processes. First, let us start with free radical scavenging. So free radical is nothing but a molecule with an unpaired electron in its outermost orbital. Because of this unpaired electron, our free radical will become very reactive and very much notorious. It will be trying to snap the electrons from the other compound and in turn can affect the entire body. Okay, so the major free radicals present in our formed in our body are superoxide radical, hydroperoxy radical and hydroxyl radical. Among these radicals, the superoxide radical formed in our body can be scavenged. That means it can be neutralized by an enzyme called as superoxide dismutase. So when that superoxide dismutase acts on superoxide radical, it will be converted to hydrogen peroxide. Okay, this hydrogen peroxide in turn will be neutralized or scavenged by an enzyme called as glutathione peroxidase, which requires reduced glutathione as a coenzyme for its normal activity. Okay, so for this reaction, the glutathione is kept, the required reduced form of glutathione is supplied only with the help of our NADPH, which is produced from the HMP shunt pathway or pentose phosphate pathway. Okay, so thus NADPH plays a very important role in scavenging or neutralizing the free radicals produced in our body. Okay, so through this action, it helps to maintain the RBC membrane integrity and also for maintaining the transparency of lens. Okay, so suppose if this function is not done properly, what will happen means the reverse, the RBC membrane integrity cannot be maintained, so it can lead to RBC membrane damage and hemolysis. Okay, similarly the transparency of the lens cannot be maintained, so leading to cataract formation. Okay, so this is the role of our HMP shunt pathway and the NADH produced from it in maintaining the or in scavenging the free radicals produced in our body. Okay, so apart from removing the free radicals, sometimes our NADPH can be used for producing free radicals, especially within the macrophages. So these macrophages, they will be having a specific enzyme, one of the unique enzyme called as NADPH oxidase. So this NADPH oxidase, they will reduce the molecular oxygen and they will convert it into free radical superoxide radical. So this superoxide radical in turn will be acted upon by superoxide dismutase to form hydrogen peroxide. This hydrogen peroxide in turn, in the presence of the enzyme myeloperoxidase, it will react with the chloride ions to form hypochlorous acid. So this hypochlorous acid in turn will be utilized for killing the bacteria which are engulfed by the macrophages. Okay, so thus NADPH plays a very important role in the bactericidal activity of macrophages. Okay, so to sum up, NADPH has dual role in the free radical metabolism. Okay, so on one hand, it produces free radicals within the macrophages. On the other hand, it helps to remove the free radicals, especially within the RBCs. Okay, so next, coming to the next function, apart from removing free radicals from our body, NADPH is also needed for removing various toxic compounds and drugs from our body. Okay, so it plays a very important role in detoxification process. So there are many different enzymes and reactions that leads to detoxification process, but one of the most important enzyme among them in phase 1 detoxification reaction is cytochrome P450. Okay, and for that cytochrome P450, one of the important coenzyme is our NADPH, which is produced through the HMP shunt pathway. Okay, so this NADPH helps cytochrome P450 and helps in the detoxification of various drugs which we take in our day-to-day life. Okay, so almost 75% of the drugs which we consume and it undergoes metabolism through this cytochrome P450 enzyme only, and for that we need NADPH, which is produced from the HMP shunt pathway. Okay, next, apart from removing the free radicals and toxic chemicals in our body, the same NADPH is also needed for removing or clearing the methemoglobin that are formed in our body. Normally, in hemoglobin, iron will exist in ferric state. Only then it can bind to the oxygen. But due to the metabolism oxidation happening in our body, sometimes the hemoglobin, the ferric ion can be oxidized, ferrous ion can be oxidized and it can be converted to ferric ions. Fe3 plus can be formed. Okay, so once Fe2 plus is converted to Fe3 plus, such hemoglobin, we call it as methemoglobin. Okay, so what is the problem with this methemoglobin means, once the ion has become Fe3 plus, it can no longer bind the oxygen. Okay, so that means it cannot, the hemoglobin cannot do its function. The main function of hemoglobin is to transport oxygen from lungs to peripheral tissues. Okay, so that function itself will be hampered when there is excess methemoglobin formation. Okay, so in our body we have a defense mechanism which immediately converts most of the methemoglobin formed in our body. We have many different enzymes. Actually, the main enzyme is actually methemoglobin reductase. For that we have many different coenzymes. One such coenzyme is NADPH. We have many different isoforms of methemoglobin reductase which uses various coenzymes like NADH, cytochrome B5, etc. like that. But this NADPH dependent methemoglobin reductase utilizes our NADPH, which is produced from HMP shunt pathway for converting methemoglobin to normal hemoglobin. Okay, the ferric ion, Fe3 plus ion in the methemoglobin will be reduced and it will be converted to Fe2 plus ion, so that it will be converted to normal hemoglobin and in turn it can be utilized for oxygen transport. Okay, next, finally, the last function is it is also needed for nitric oxide synthesis. Okay, so nitric oxide, how it is synthesized? It is actually a potent vasodilator produced in our body from the amino acid arginine. Okay, so arginine by the enzyme arginase, by the enzyme nitric oxide synthase, it will be converted to nitric oxide and citrulline. Okay, so this enzyme nitric oxide synthase, it needs many coenzymes. Out of that, one of the important coenzyme is NADPH. Okay, so that's it about the functions of NADPH. To summarize, so from HMP shunt pathway we are getting two important byproducts. One is pentose phosphates and the other one is NADPH. The pentose phosphates produced will be utilized for purine and pyrimidine synthesis, which in turn will be utilized for DNA synthesis, RNA synthesis, ATP, GTP, various coenzymes and second messenger synthesis. Okay, whereas coming to the NADPH, it can be used as a reducing equivalent for reductive biosynthetic reactions. In addition to that, it also plays a direct and major role in free radical scavenging, bactericidal activity of macrophages, detoxification process within the liver, methemoglobin conversion, and also for nitric oxide synthesis. Okay, so these are some of the major functions of NADPH and pentose phosphates which are produced from HMP shunt pathway. Okay, so before winding up, I would like to give you a brief small clinical significance related to this pathway. Okay, so one of the important enzyme, actually the key, the rate limiting enzyme of this pathway is G6PD, glucose 6-phosphate dehydrogenase enzyme. Okay, so it is actually in some condition in this enzyme may be deficient. Okay, if this is deficient, what will happen? NADPH and pentose phosphates may not be produced in sufficient quantities. Under such conditions, what will be the main symptoms the patient will be getting is hemolytic anemia, neonatal jaundice, and methemoglobinemia. Okay, the newborn baby born with this congenital disorder, okay, hereditary disorder, will be usually suffering from, will be presenting these three clinical features. So why there is anemia and jaundice means due to excessive hemolysis. Why there is excessive hemolysis means due to defective free radical scavenging process within the RBC, which in turn is due to decreased production of NADPH, which in turn is due to the deficiency of the enzyme G6PD. Okay, so apart from decreased free radical scavenging, when NADPH is not produced in enough quantities, even the methemoglobin conversion back to hemoglobin will also be affected, and the children may be presenting with methemoglobinemia also. Okay, so normally most of the G6PD deficit is actually X-linked recessive disorder. So actually most of the patients will remain asymptomatic only. Okay, so these patients, when they are precipitated by some triggering factors like some oxidative the drugs which increases the oxidative stress in our body, like primaquine, sulfur drugs, fava beans, and also certain infections. Okay, so all these things, they will increase the oxidative stress within the RBC. At that point of time, the defective G6PD enzyme cannot produce enough quantity of NADPH, and that point they will be manifesting with hemolytic anemia and jaundice. Okay, so that's it about the various significance of HMP shunt pathway. Thank you.